Alright guys, here we are for concept 15.4. This entire chapter, chapter 15, is about tying what we physically actually see during meiosis, mitosis, and that type of thing. What we physically see the chromosomes doing in the actual physical chromosome that we see within the microscope, tying all that back to the ideas, the theory of Mendel and Morgan and all of these others. So we're tying physical evidence back to the various theories that the information and evidence that was collected kind of helped support. So what Mendel did was he did all of these various experiments, and he came up with a hypothesis that was supported by his data. And then later we took physical data, physical examples, physical evidence, and that also helped to support the same theories and ideas that Mendel developed as well. So chapter 15 is all about physical evidence for our thoughts and various scientific ideas regarding heredity and genetics. The last one here says concept 15.4, alterations of chromosome number or structure cause some genetic disorders. So again, we're going to look at a physical occurrence where the number of chromosomes is incorrect or there is something physically wrong with the chromosome. How does that affect what we see? How does that affect phenotype and what goes on in the body? So it says we've already seen how minor changes to individual genes can affect the phenotype of an organism. So we have different alleles for a trait. So an allele, let's just pick one like for pea flower color. The change between a white flower and a purple flower, the allele for flower color, the difference there may only be a nucleotide or a couple of nucleotides. It's a very minor change between those alleles, but it ends up showing completely different flower colors. So that's a very small change. What if we have a large change, large-scale chromosomal alterations in humans and other mammals? Oftentimes lead to very bad things. So very often, if you have large-scale chromosome changes, it's going to lead to a spontaneous abortion or miscarriage of that embryo before it ever really gets too far in its development. So if there's something majorly wrong with the fertilized egg, with what came from the sperm or what came from the egg, before that embryo tends to get very far along at all, it will spontaneously be aborted because there is something so wrong with the genetic information that it doesn't allow for proper development. If it does allow for development through a certain stage, you could have major developmental disorders or other problems if that offspring is able to survive to birth, if that embryo develops into a fetus and then into a viable offspring. Now, plants seem to be a little bit better at tolerating these genetic changes than animals. We're going to see where in plants some of these what would be very detrimental or lethal to an animal embryo, actually the plant actually is able to survive and grow with that change. But in animals, usually large chromosome changes are deadly. So let's take a look at some of the possibilities. The first possibility that something might go wrong is where you have an abnormal chromosome number, where you don't have the right number of chromosomes within the cell. And usually during the whole lining up and separating, where the meiotic spindle separates and pulls the chromosomes toward one particular end of the cell, that usually happens without error. So, that should say without, not without. That usually happens without error, but sometimes problems do arise. One of the possible problems is called non-disjunction. Non-disjunction is where pairs of homologous chromosomes don't separate normally during meiosis 1. So during meiosis 1, when those homologous chromosomes line up in the center and then they get separated from each other, if that doesn't occur, you're going to have a problem because both sets of the homologous chromosomes are going to be pulled to one side. Or if the sister chromatids fail to separate in meiosis 2. So again, we're talking about meiosis because we're talking about the creation of gametes, which is the reproductive cells. So in meiosis 2, remember you have the sister chromatids that are then lined up, and then they're supposed to split apart and separate. So you have the individual chromosomes going to either side. Well, if for some reason that doesn't happen, you're going to have a duplicate set in one side and no copies in the other. So as a result, one gamete may receive 2 of the same type of chromosome, and another gamete may receive no copy of that chromosome. And then that may only affect one chromosome with all the other chromosomes being distributed normally, but that's still going to be problematic. So here are your two possibilities. In meiosis 1, so let's look on the left first. In meiosis 1, here's a non-disjunction where we had the homologous chromosomes lined up and they were supposed to be separated from each other, but they weren't. They were both pulled to this side. And then during meiosis 2, you have everything occurred normally, but by then you already have multiple copies in this one. And so here are your 4 gametes. This one ends up with multiple copies of this chromosome, and then these two end up with no copies of that chromosome. So that's non-disjunction of the homologous chromosomes in meiosis 1. How about the other side? Let's say meiosis 1 works out fine, they separate the way they should, but then in meiosis 2, where the sister chromatids are supposed to pull apart and separate, that doesn't happen right here. You have a non-disjunction. So in these 2 gametes that are created from this cell, they have an unequal distribution, where in this one there is 2 copies of that chromosome, and this one has none. These 2 gametes were unaffected because this process occurred normally, so these 2 were unaffected. So these are the 2 possibilities for non-disjunction, where the chromosomes don't separate the way they should. So if that occurs, you're going to have some gametes that don't have enough chromosomes and some gametes that have too many. And if one of those gametes is fertilized, if it is part of the fertilization process where non-disjunction occurred, you're going to have something called aneuploidy. So aneuploidy is the fertilization of one of these gametes that has an improper number of chromosomes. And so the offspring with this condition had an abnormal number of a particular chromosome. They can either be called monosomic. It's a monosomic zygote, and it only has 1 copy of a particular chromosome instead of the normal 2. Or it can be trisomic. Trisomic has 3 copies of a particular chromosome. So monosomic would have 1 too few. Trisomic would have 1 too many. Down syndrome is a good example of trisomy in humans. Down syndrome is the result of a trisomic zygote. So that zygote has 3 copies of one particular chromosome instead of the normal 2. Aneuploidy can involve more than 1 chromosome. So generally you would think about this problem only occurring with 1 chromosome, but sometimes it happens with more than 1 chromosome. And after fertilization, mitosis will then transmit this problem to all of the embryonic cells created. So remember, you're starting out with a single fertilized egg from the combination of the male sperm and the female egg. But if that fertilized zygote, that fertilized egg, then begins to create new cells through mitosis, but remember, those cells are all daughter cells that will be genetically duplicate. So if that beginning cell is monosomic or trisomic for a particular chromosome, well then all of the cells afterwards will also have that particular problem. Now non-disjunction can also occur in mitosis. So you can have this occur later on. So that embryo is developing. Let's say that embryo is now 5 cells, 10 cells, 100 cells, and one of those dividing cells through mitosis has this problem. Well, then all the future daughter cells of that one will also exhibit this problem. Now, if this happens really early on, well, then you're going to have a huge percentage of the cells affected by this, and the whole organism is likely to exhibit substantial effects and problems. But if you had this happen much later in the development, it might not be as severe because only a small percentage of all of the body cells will be affected. What if we're not talking about just a single chromosome? What if we're talking about the entire set of chromosomes? There's something called polyploidy. Polyploidy, poly meaning many. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. So as if there were multiple parents somehow involved, but really it's just a duplication of the chromosomes given by the 2 parents. So more than 2 complete sets of chromosomes. You can have triploidy, 3N, which is 3 times the normal amount, 3 sets of chromosomes. Tetraploidy, which is 4 sets of chromosomes. Polyploidy is pretty common actually in plants, but not in animals. This would almost always be uncommon, perhaps lethal or detrimental situation in animals. But polyploidy is actually fairly common in plants. And it might sound unusual, but polyploids are usually more normal in appearance than aneuploids. So it's better to have a full extra set of chromosomes than it is to have 1 extra or 1 missing chromosome out of the entire set. So one extra or missing chromosome seems to have more of a disruptive effect than does having just a full entire set. So it's better to have a duplicate of the matched whole set than it is to have one out of that entire set be wrong or incorrect. Some of the common plant polyploids include bananas. Bananas are triploid. They have 3 complete sets. Wheat is hexaploid. It has 6 complete sets. And strawberries are octoploid. They have 8 complete sets of the genetics inside of them. So you can see in plants, there's a lot of varieties of plants that for them, this is kind of what you would consider the normal, but that's not the case in animals. What if we're not talking about having an incorrect number of chromosomes? What if we actually do some damage, physical damage to the chromosome itself? So errors or breakages in the chromosome, this is going to be some sort of copy error in meiosis, or can be caused by damage from the environment, such as radiation. That can lead to 4 types of changes in the structure of the chromosome, the physical structure. The first one is called deletion, and that's where a piece of the chromosome is removed, a segment of the chromosome is removed. The next one is called duplication. That's where a section of the chromosome is duplicated, copied, repeated. The next one is inversion. That's where a piece or a segment gets reversed in its orientation. And the last one is translocation. This is where a segment moves from one chromosome to a different non-homologous chromosome. So a section of the genetic code basically jumps from one chromosome to a completely separate non-homologous chromosome. If a portion of the DNA or the gene, the chromosome, goes through this process, well, you can have different effects. If the centromere is deleted or the centromere is disrupted, usually the entire chromosome is going to be destroyed or lost. The lost or deleted fragments of a chromosome can be attached as an extra segment to a sister chromatid or a non-sister chromatid of a non-homologous chromosome. Or that fragment could become reattached to the original chromosome, but in reversed order, or it could go attached to a different chromosome. So you've got all these 4 possibilities. So let's say a piece of the chromosome breaks off or gets removed. That's called deletion. Let's say that it goes and attaches itself to the sister chromatid. Well, then you might have duplication. Well, let's say that segment reattaches back where it was, but in the reverse order. Well, that's called inversion. Or let's say that section goes off and reattaches or attaches to a whole different chromosome. That's called translocation. So you have all these possibilities of what might happen when the chromosome gets broken or damaged. Great illustration in your textbook here about some of the different possibilities. So you have deletion, where a portion is removed, a portion of that chromosome gets lost. So here you see the D portion of the chromosome has been deleted. Then you have duplication. Duplication is where there's a repeat. So see the BC region was duplicated in this chromosome. Here you have inversion, where this portion somehow became removed and then reattached, but it was inverted or reversed. And then you have translocation, where a piece of one chromosome simply is removed or dislodged and goes and attaches to a completely separate, different chromosome. These are all the 4 possibilities. Deletions and duplications, these 2, are especially likely to happen during meiosis. In crossing over, sometimes the non-sister chromatids swap an uneven amount of genetic material, and that results in a deletion from one and a duplication in the other. So if the sister chromatids are crossing over and they're swapping genetic information like we've talked about, but if they don't swap the same amount, well, then in one of them you're going to end up with a deletion. It didn't get as much as it needed. And in the other one you're going to end up with a duplication. It got a little too much. So large deletions are usually lethal. That's usually going to cause the death of the developing organism. But some of these other changes can also cause either the death of the developing organism or possibility of different problems down the road. The last thing we want to look at is some of the human disorders that are caused by these alterations of your chromosomes. So this would be, again, either a change in the number of chromosomes, either you have too many pair or too few, or some sort of change in the structure. Usually it's going to be the cause of some serious disorders. Some types of aneuploidy, remember aneuploidy is where you don't have the proper number of chromosomes. Some types of aneuploidy appear to upset the genetic balance less than others. So some of these result in the death of the developing embryo, some of them do not. Some of them, the embryo can continue to develop and the baby can be born. But this usually results in some sort of problem down the road. But the individual can survive, be born, and sometimes live a fairly long, good life. So these surviving individuals usually have a set of symptoms that we call a syndrome, and that's usually characteristic of some type of aneuploidy. Probably the most common type of aneuploidy is something that develops into what we call Down syndrome or trisomy 21. And it's called trisomy 21 because the 21st pair of chromosomes, where remember humans have 23 pair, well, pair number 21, there is a third chromosome instead of the normal pair of 2. Unlike many of the aneuploid conditions that result in the spontaneous abortion of the embryo or the death of the fetus, those with Down syndrome can usually lead pretty lengthy, meaningful lives. They are born and they can be fairly healthy throughout their entire lives and live a good long life. Common symptoms of Down syndrome usually include kind of a set of features that are recognizable characteristic facial features, short stature. Sometimes the children are born with heart defects, but are often correctable heart defects. Some developmental delays and a somewhat shortened lifespan sometimes are some of the results of this. So Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21. That's why it's also called trisomy 21. It affects about 1 out of every 700 children born in the U.S., and the frequency of Down syndrome actually increases with the age of the mother. And that hasn't been fully explained yet, but the older a woman is when she has her children, the greater the chance that one of the children will be born with Down syndrome. And although this is an area of study, we don't know exactly why that's the case, but some of the experiments that have been done, some of the study that's been done, says it's often a result of non-disjunction in meiosis 1. So remember in meiosis 1, when those homologous chromosomes are separating, you might have a problem there. And some of the research says that there's an age-dependent abnormality in a meiosis checkpoint that delays anaphase until all kinetochores are attached to the spindle. So what that means is, as the woman ages, sometimes her cells have a slight abnormality where as all of the chromosomes are lining up in the center, getting ready to separate, well, there should be a checkpoint that tells the cell, okay, wait until we're all connected, until all the kinetochores are connected and attached to the spindle, and everything's lined up, and then pull apart into anaphase. Well, if that checkpoint doesn't stop the cell from pulling apart into anaphase, not all the chromosomes may be lined up and properly connected to the kinetochores, to the spindle, and then you're going to have a problem. So here's a karyotype of someone who would have Down syndrome, and you can see right here, chromosome set 21, there are 3 chromosomes instead of the normal pair. Here's a cute little young girl who has Down syndrome. And again, luckily this particular syndrome is not usually lethal, and these children can grow into healthy and happy people. Aneuploidy can occur for the sex chromosomes as well. So like with Down syndrome, it was look at the karyotype, the number 21 set of chromosomes that has the incorrect number. But aneuploidy can happen with the sex chromosomes too, so non-disjunction of the sex chromosomes, that can produce a variety of these aneuploid conditions. But oftentimes, if you have non-disjunction of the sex chromosomes, it appears to not upset the genetic balance quite as much as when this happens to the autosomes. So oftentimes, you won't even see any results of this, or you won't see any major detrimental effects as you would when you have aneuploid conditions of the autosomes. The reason that aneuploidy of sex chromosomes probably doesn't cause quite as many problems is the fact that the Y chromosome carries very few genes on it, so there's less likely of major changes or damage to occur because you're not talking about that many genes. And then remember, the extra X chromosomes are going to be inactivated into bar bodies in the somatic cells. So if you have too many of the X chromosomes, well, the extra ones are going to be inactivated usually anyway, so you wouldn't see some of the major problems and effects. There's more information about some of these problems and effects on page 299 and 300 of your text, but we'll talk about just a few here. One of them is called Klinefelter syndrome. That's the result of an extra chromosome in a male producing XXY. So instead of the normal XY, you have a problem that occurred in the egg production of the female. And so there were two X's within the egg, and then, of course, the Y came from the sperm cell. So this is going to be a male, but they have an XXY sex chromosome set. These individuals will have the male sex organs, but the testes are usually abnormally small, and the man is usually sterile, and there can be some other physical traits that are affected by this as well. An example in females is called monosomy X, also called Turner syndrome, where the female is basically XO. In other words, she has the X chromosome, but she did not receive either the other X or the Y chromosome. So these are females. They'll have the usual female body parts, but they're generally sterile, and this is the only known viable monosomy in humans. So any other monosomy in the autosomes would result in the spontaneous abortion of that embryo, but this can result in a live birth, and generally the people who have this condition, they'll take a variety of injectable hormones, and that will allow them to live a pretty normal life with all the various female traits that you would normally expect. Some of these do not result in a major problem, like, for example, there is a possibility of being trisomy X. So a female who is trisomy X with 3 X chromosomes, you probably would not even really know it. They are generally healthy and have no unusual physical features, other than oftentimes they seem to be slightly taller than average. But for the most part, you might not even know that they had any sort of genetic issue. So there's a fairly wide variety of conditions, and you can have aneuploidy of the autosomes, or you can have aneuploidy of the sex chromosomes as well, and that can sometimes be lethal, detrimental, or it can allow for the embryo to eventually develop and be born, but there's oftentimes characteristics and syndromes that go along with that. There are some human disorders that are caused by structurally altered chromosomes. These are usually pretty detrimental. Oftentimes, if there is a structural change in the chromosome, a deletion or removal of a portion of a chromosome, that's going to result in the spontaneous abortion of the embryo. But on occasion, the embryo will continue to develop. One of them is a very odd one, an interesting one called cri du chat, which is French for cry of the cat, and it results from the specific deletion of a portion of chromosome 5. And children born with this syndrome have mental retardation. They have some particular facial features, characteristics, and when they're very young, their vocal cords and airway is not fully developed, and that gives them a very cat-like cry. They sound like a crying kitten or a mewing cat, so that's where it got its name. These individuals sometimes die in infancy or early childhood, but on occasion they're able to live with proper treatment into young adulthood. There are also certain cancers, including one called chronic myelogenous leukemia, CML, that's caused by the translocation of chromosomes, where a piece of a chromosome is taken from a gene, or a piece of a chromosome is taken from one portion and moved to a completely non-homologous different chromosome. So many deletions of chromosomes are lethal or cause severe problems. Translocations tend to also be pretty problematic. This CML leukemia is caused by translocation in mitosis of the cells that are going to become white blood cells. So there becomes a problem in the cells that go on to become white blood cells, and that leads to the activation of a gene that causes uncontrolled cell cycle progression. So that breaks down that whole cell cycle where the cell keeps reproduction, mitosis, in check, and that leads to tumors and cancer. In the chronic myelogenous leukemia, the CML, what's happening is a translocation between chromosome 9 and chromosome 22, where you have pieces of these chromosomes actually swapping onto different non-homologous chromosomes. And that causes this particular chromosome, which can be shown in the karyotype. It's called the Philadelphia chromosome. I'm guessing it's because perhaps the lab or the doctors who first noticed this or located are located in the city of Philadelphia. But that chromosome, the translocated chromosome 22, is of a slightly different shape than it should be. You notice it's quite a bit shorter, and you can see that if you do a genetic karyotype, and that can help determine what's causing the problem that you might see in the person. All right, we're going to wrap it up. The last concept for this chapter. We have talked about a lot of laws and rules of inheritance that Mendel helped develop, and of course we've seen where there can be problems that arise. But now we're going to talk about some inheritance patterns that are the exception to the standard Mendelian inheritance. So we're going to talk about, in biology in particular, there seem to be rules, and if there are rules, there's going to be exceptions to those rules. Life can be pretty messy, so you have to be flexible when you're studying biology. So we've discussed things that can go wrong, abnormal occurrences, but then there are also two normal exceptions to Mendelian genetics as well. One exception involves genes that are located in the nucleus where you would expect, and the other exception involves genes that are located outside the nucleus. Did you forget about those? Remember, there are DNA strands in other parts of the cell inside the mitochondria and the chloroplast. So in both cases, the sex of the parent contributing an allele is a factor in the pattern of inheritance. So we're going to look at two inheritance patterns that don't follow the usual rules. One of these involves genes in the nucleus. One of them involves genes located outside of the nucleus. And in both cases, the sex of the parent has a factor to play in how these are going to be inherited. The first exception we're going to look at is called genomic imprinting, and this is regarding genes that are located along with the vast majority of the genes in the nucleus of the cells, but it has something to do with where those genes come from, whether or not they came from the mother or the father. In general, it makes no difference if a particular allele came from mom or dad. Say, for example, in Mendel's pea plants, the allele for white flowers or the allele for purple flowers. It didn't matter if the purple flower allele came from the mother or the father, it had the same effect. And that's true for the vast majority of the genes that we're looking at here. But for a few mammalian traits, the phenotype, what you see, actually does depend on which parent, the male or the female, passed along the particular allele for those traits that we're looking at. Now, these are not like the sex-linked genes necessarily that we were talking about earlier, because most of these genes, these imprinted genes, are located on autosomes. So even though the sex does play a part, or actually not the sex of the offspring, but the sex of the parent that the allele came from. So in the sex-linked genes, oftentimes it is the sex chromosomes that came from the parents, and then the sex of the offspring that determines some of what we're going to see. But in this case, we're talking about genes that are normally located on the autosomes, and the alleles that mom and dad give, and how they differ based on whether or not they came from the male or the female, the mom and the dad. This type of variation in phenotype is what we call genomic imprinting. And the way it works is that the imprinting basically involves the turning off or silencing of certain genes that are stamped with an imprint during gamete production. So let's take a look at an example. One of the very first genes that was discovered to have this genomic imprinting possibility was a gene in mice, and this was a allele that produced a particular protein. The gene would code for a particular protein, and it was a kind of an insulin-like growth hormone, so an insulin-like growth factor. So they've called it IGF here in this picture. So here at the top, you have a mouse that is homozygous for a particular trait. It has the normal IGF trait allele from the father, the paternal side, and the normal IGF trait allele from the mother side. So it's homozygous for that, and that produces this normal-sized mouse, the wild type that we see here. In this case, you would have no idea that there was any sort of genomic imprinting going on, because this offspring mouse received the normal allele from dad and the normal allele from mom. So that would tell you basically nothing. But what if you have two different mice and they are heterozygous for this trait, where they received one of the normal alleles and one of the mutated alleles? The mutation causes a dwarf mouse, a smaller sized mouse. So here we have two different mice, two different offsprings, and they are heterozygous. In this case, this mouse got the normal gene from dad and the mutant gene from mom. This mouse got the mutant gene from dad and the normal gene from mom. Now, if there was no relationship between the sex that the allele came from and there was no genomic imprinting, you would expect both of these to look the same. Both of these mice are heterozygous for the trait, but you would expect both of them to look normal because you would expect that that is the dominant trait and the normal size is the wild type. But take a look at what you actually see. That's not what you actually see in this case. What you see is where the mouse received the mutant allele from the mother, that's not seen. The normal is what you see. The normal is expressed. Over here, what you see is the mutant was received from the father, and that's what shows up. Well, why would that be the case? Because this mouse also has the normal from the mother, and you would think that would be dominant and it would overtake the recessive mutant allele. Well, that's part of this genomic imprinting. In this case, the genomic imprint is such that the maternal version, the mother version of the allele, is inactivated, and only the paternal allele is what gets expressed. Now, you wouldn't know that up here because this mouse has both the normal from mom and dad. So if mom's allele is inactivated, it doesn't really matter. Dad's allele shows you the normal size. Down here, this mouse has normal from dad, mutant from mom. Now, you wouldn't really expect to see that either because, again, you would still think that normal is dominant and it would overtake that. That's what you would see. This is the one that's interesting because, again, you received one normal dominant allele from mom, one mutant recessive allele from dad. And again, you would expect this mouse to look the same as this one because they're both heterozygous for that trait. But here's the genomic imprinting. In this case, with this allele, all of the alleles from the mother's side are inactivated, turned off. So the mother's normal allele has basically been switched off, inactivated, and so the recessive mutant allele can show up and you have a dwarf mouse. Now remember, this is not related to the sex of the offspring. These are on the autosomes, so this could be a male mouse or a female mouse, but it's the mutant trait that shows up because it's located on the allele, the chromosome that came from the dad, whereas the mother's allele was inactivated for this trait. That's genomic imprinting. The chemistry behind this is sort of interesting. Excuse me. The chemistry behind this is sort of interesting. It appears that this imprinting, this turning off of the particular allele, is a result of what we call methylation, the addition of a methane molecule that has a methane molecule attached to CH3 of cysteine nucleotides. And so the addition of this cysteine nucleotide, this silences that maternal allele or that maternal gene that came from the mom. Now that could be from the father. So there are others that are genomically imprinted where if the allele comes from the father, it's silenced. So it's not always the mother, but just in the mouse case it was. While genomic imprinting is thought to affect only a small fraction of mammalian genes, scientists only know of maybe a couple of dozen of mammalian genes that are like this. Most imprinted genes are critical for embryonic development. So while there may not be very many of them, they are pretty important for the early development of the embryo. And they have done quite a few experiments where they modify this imprinting. And what they found is that most of the research shows that the developing zygote has to have one active copy. If there was a way, say for example in our mouse example, to turn on both the alleles from the mom and the dad, oftentimes that resulted in a problem. So if there were two copies, or if they inactivated both copies, the scientists found that oftentimes that caused problems. So with most of these imprinting genes, this genomic imprinting on those genes, the developing embryo has to have one active copy, not 0, not 2, has to have the 1 to develop normally. So while this is not common amongst all the genes in mammals, there are a few that whether or not the allele came from father or mother does have an effect on what we see in the phenotype. So that's one example of some inheritance that's not going to follow the normal Mendelian rules that Gregor Mendel was studying in his pea plants. Because for most of the genes, it doesn't matter if the allele comes from mom or dad, it has the same effect, but in this case, that's not the case. The last ones we're going to look at are the genes that are not in the nucleus. Some of the genes that are located in other parts, they're called extranuclear genes. Those are going to be cytoplasmic genes, that's another word for them. They're found in the organelles, in the mitochondria, the chloroplast, and plants have some other organs called plastids that are kind of similar to chloroplasts. Chloroplasts are a type of plastid, and they contain separate small circular DNA molecules. Remember, there was the theory, or there is the existing theory, that eukaryotic cells developed from prokaryotic cells that engulfed other living cells. And so perhaps the mitochondria, the chloroplast, were their own separate living creature at one time, and they were taken in and became organelles through the process of evolution over time. That's why perhaps they have their own separate strands of DNA. So extranuclear genes are inherited maternally because the zygote cytoplasm and all the associated organelles come from the egg. So when you have a fertilization occur where there's a combination of egg and sperm, there's not a whole lot there in the sperm. The sperm is basically just the set of genetic DNA, and there's not a lot of other associated material. But the egg is quite a bit larger, and it contains cytoplasm and organelles and everything else that's going to get everything going for that first initial cell. So when that zygote first begins to grow and then divide, well, it has to have mitochondria and endoplasmic reticulum and everything else. Well, that came from the egg. So it was the mother that provided all of that material. So these extranuclear genes that would be located inside the mitochondria, that always comes from the mother. Or in the case of plants and the chloroplast, that always comes from the mother, basically. The first evidence that extranuclear genes came from studies on the inheritance of yellow or white patches on leaves of otherwise green plants. And I'll show you a picture, and you've probably seen this type of thing before. So this is what we're talking about. This is called variegated. So if you have plants like this, maybe your mom or dad have houseplants like this that are called variegated, where normally these leaves would be nice solid green, but you have these patches or variations of yellow or white or other colorations. That's the trait that we're kind of looking at. And this trait was helpful in illuminating this this genetic material that was not located in the nucleus, located, say, in the mitochondria or the chloroplast. In this case, it's probably going to be in the chloroplast because we're talking about plants. Let's find out. So when I look up this information about the plants, what it says is the coloration that we see, this yellow and this mottling color, this variegation, these are called variegated leaves. The coloration is caused by mutations in the plastid genes that control pigmentation. So remember I said plastids are a type of cell organelle within plants, similar to chloroplasts. And in those plastids there is separate DNA, separate from the nuclear DNA. And if that DNA has a particular mutation, that can affect how pigment is laid down within the leaves. So plastids are a type of organelle structure found in the plant, contain their own DNA. An egg may contain different plastids with different alleles for coloration. So you can have a variety of plastids within the egg that have different DNA from each other. The DNA within the plastids can be different. And sometimes in some of those plastids, the DNA has this mutation that changes the coloration. As the plant embryo starts to grow and then divide and undergo mitosis, so you're going to have some of those daughter cells that get the plastids with the mutation. Some of those daughter cells are going to get the plastids that do not have the mutation. So some of the cells in the leaves will then have that mutation. Some of the cells will not. And what you're going to see is this mottled effect right here. So say, for example, the yellow is a mutation. So these cells that grew from mitosis, dividing, they have the plastid that has the mutation in its DNA. These cells with the green do not. So the cells of this plant, the cells of the leaves, are made up of cells that have different DNA within the plastids of the plant. You can also see some of this type of inheritance, maternal inheritance, coming from the mother again, because the egg is what provides the organelles and the cytoplasm and everything inside for that beginning embryo. So similar maternal inheritance is also the rule for mitochondrial genes, genes located inside the mitochondria in most animals and plants, because almost all the mitochondria passed to the embryo comes from the egg. Again, the sperm really doesn't provide anything but a haploid set of chromosomes, but all of the cytoplasm and organelles tend to come from the much larger egg. Some defects in mitochondrial genes will prevent the cell from making enough ATP, and that can result in some diseases that affect your muscles and your nervous system. There's a couple of problems, one of them called mitochondrial myopathy and one called Leber's hereditary optic neuropathy. These are all related to the genes located inside the mitochondria, which are different, separate set apart from the nucleus. The product of most mitochondrial genes. What do most of the mitochondrial genes do? Well, they are instructions for making proteins, the various protein complexes of the electron transport chain and the ATP synthase. So remember, inside the mitochondria, that's our powerhouse of the cell. That's where we have that electron transport chain, and we have that little pump that we call ATP synthase to create ATP. So if you've got a problem with the genetics of the mitochondria, you might have a problem creating the proper proteins that you need. So those defects are going to lead to problems in the proteins, and your cell might lack the ability to create the ATP that it needs, the energy that it needs. Finally, just to finish off this chapter, you may have heard of this term mitochondrial Eve, and scientists study the DNA inside the mitochondria quite a bit because it's a direct linkage. It's passed down from mother to offspring, from female to female to female in a direct line because of the way that this DNA gets inherited, and because the egg is what provides the mitochondria, and that is not mixed with anything else. The DNA inside the mitochondria or the organelles is not part of that fertilization process where those genetics are mixed with the father. So there's this idea and this perhaps a story that you've read about mitochondrial Eve. It's what it's called. It's a really interesting thing. I suggest if you have the time to look it up. I basically just took this information off of the Wikipedia page, and there's a link right there. But in the field of human genetics, they have used this term mitochondrial Eve to refer to the matrilineal most recent common ancestor of all currently living modern humans. So you can go through the genetics of most people from around the world, and it will lead you back by looking at the mitochondrial DNA to one female that at one time existed way back in our history at some point, approximately 140 to 200,000 years ago, and that is the most recent woman from whom all living humans are currently descended today. Now, I don't want you to think that means that was the mother of everyone or the beginning mother. That's not exactly what that means. It simply means that she is the most recent woman in our history from which our mitochondrial DNA for all the currently living people came from. That doesn't mean she was the only living female human at her time. All that means is because of the way that the mitochondrial DNA is passed down, she is the only female or the most recent female in previous history who has a continual line of female offspring. So there could be a mother who lived at the same time who didn't have a daughter, who only had a son. Well, then that breaks that linkage, because that son, he's going to pass on his sperm to his children. He's not going to pass on an egg. So this only works with the female side of the equation. So a mother having a daughter who then has a daughter who then has a daughter. So you can back that path all the way up to this mitochondrial Eve. So kind of a cool story, interesting information. Check out the Wikipedia article if you get the chance. All right, so this entire chapter, let's wrap it up, is all about the physical aspects of the cell and how we can combine chapters 14 and 15, the information that we learned from Mendel and our words and our thoughts and our ideas, and how does that correspond to what we physically see? What is our physical evidence for that inside the cell and the chromosomes themselves. So our next chapter is basically about DNA. That DNA molecule has to replicate, has to divide, and then prepare itself to be split in two. So how does the DNA molecule replicate itself in preparation for all of this division and reproduction? Well, that's part of the next chapter. So we're going to stop right there. Chapter 14 and 15, I don't think the information is difficult, but I think there's a lot of it. So I would highly suggest that you read over it, take a break, come back, really go over and look at the chapter summaries, because they combine into 1 or 2 pages all the information that you need, and then just kind of go over it multiple times. Again, 14 and 15, lots of good information, but I don't think it's particularly difficult information. There was just a lot of reading involved. Do the study sessions online. Make sure you ask me any questions. Go watch the videos. Take advantage of all the possibilities for looking at this information, and I think you'll do much better. Have a good day. We'll talk to you soon. Bye-bye.